Okay, question number 5. Am I recording? Yes, okay. Question number 5. So name sections 1, 2, 3, and 4. So this is a leg operon. The question says this is a leg operon. So an operon will contain 3 sections, right? Which is POS, correct? So therefore, number 1 is the promoter. It's my pencil line. So this is promoter, this is operator, and all these are the structural genes. So number 1 is promoter, number 2 is operator, and number 3 is structural genes. Here. Number 3 and 4 different. Number 3 and 4 is the name of that particular gene. So the name of the particular gene is one. Where's my mark scheme? Here. Paper 4. So question 5. Okay. So that means you need to memorize. You need to memorize the whatever name you have learned in your syllabus. Please memorize them. Okay. LACZ, coding for beta galactosidase gene that breaks down lactose to glucose and galactose. Y is lactose permease gene, the gene coding for this enzyme. So permease means what? Making the membrane permeable. So it's a coding for a enzyme that catalyze the synthesis of the carrier protein for lactose. Okay, because why? ACE means it's an enzyme coding for the protein, carrier protein. Okay, next question, part 2. So they say that gene I is located in a short distance away from leg operon. The product of gene I, which is actually the repressor, right? So the repressor is a constitutive protein. So the question says suggest means you have to think. Suggest means whatever you have learned that you can write here. So they say that suggest what is meant by the term constitutive protein. So you learned before that when you learned leg operon, this gene will code for a repressor protein. Okay. They will always be synthesized. And this repressor protein, when synthesized, it will bind to, so this is repressor protein. It will bind to the operator, and therefore it blocks this region so the RNA polymerase cannot bind. So it's always synthesized. So therefore the answer is, so this is whatever you have learned, right? So the answer is, yeah. Protein, okay, that is made all the time. Protein that is made all the time. So this is what you have learned, isn't it? So you write whatever you have learned, okay? Okay, next question. Part 3, It says that a strain of E. coli has been produced with a mutation in gene I. So the gene I has been mutated so that when the gene is expressed, it will still produce protein after mutated. But because the base sequence of the DNA changed, so the base sequence of mRNA changed, amino acid sequence changed, the protein structure will change. So when the structure change, function will change. So it says that it's no longer functional. That means it can no longer bind to the operator. Okay, no longer bind to operator to prevent the transcription of the gene, right? So it can no longer bind to here. So cannot bind to here anymore. So when it cannot bind to here, yeah, what is this? Okay. So cannot bind to here. So therefore, the RNA polymerase can bind and start transcription. So they ask you what happened. when that repressor protein is mutated, right? So you cannot bind. So all these leg Y, leg Z, and leg A will always be expressed. So when it's always expressed, whether lactose is present or not, it will still be expressed. Okay. So lactose present or absent, we will still express the enzyme. It means it's a waste, isn't it? Because when we don't need the enzyme, we still produce the enzyme to break down lactose, to transport lactose into the cell. So it's a waste of energy. So you look at the mark scheme, it says that where is it? So the enzymes are made all the time. It's same mark scheme as previous one. So the previous answer is also same thing, produce all the time. The previous answer. So this answer is the enzyme coded by leg Z, leg Y, leg A will be made all the time. So they will produce too much when the cell do not need, means in excess. So it's a waste of the cell's amino acids, a waste of ATP, a waste of nucleotides, because they're producing the mRNA, they're producing the protein, even though the cell, there is no lactose, okay? So that will cause a decrease in growth. That means when the cell has lack of ATP, there's lack of energy for growth. Okay. So question number 6. So it says there are 600 plant species for this flowering plant. Okay, they call it morning glory. So 600 plant species. So it says that the flower color is coded by 2 alleles. Dominant allele gives purple flower, recessive allele gives red flower. Okay, so a student record the flower color of the plant in a field, and the student recorded 660 plants with purple flowers and 440 plants with red flowers. Okay, so purple is coded by dominant allele, red is coded by recessive allele. So in this case here, recessive, so 440 plants that are recessive, so their allele must be recessive, so small p, I put small r, so recessive, okay, recessive. So for those that are purple, their genotype should be big R, or big R, small r, right? Okay, so it'll be purple. So recessive is this genotype. So this is genotype. So this is a Hardy-Weinberg question. So assuming that Hardy-Weinberg principle applies, okay, that means the phenotype remain more or less constant. Okay, so use the equation, show your working to give your answer in the nearest whole number. So you need to underline as you read the question because they want the answer to be whole number only. So you score full marks. So let's look at the question. So what is the number of heterozygous plants? So you need to use this equation to calculate, okay? P plus Q is equals to 1. So here, just now, 440 plants have got red flowers. So they are recessive. So recessive, Q square is the frequency of the small r. Okay? Q square is the frequency of small r. So let's look at the mark scheme working, so it's faster. Okay. So the Q square represents the small r genotype, right? So it's frequency of, so altogether there are 440 red flower. And then this is a total red flower plus purple flower. Okay, out of total. So Q square is equal to 0.4, but there's no marks given because it's in bracket. The mark is given if you convert the Q square to Q. That means you must root square 0.4. So your Q answer is this one. No need to round up. You don't need to round up because you still need to calculate until the last answer. So while you do the working, don't round up until the end. Okay? So after that, you want to find out what? You want to find out, what was the question? Number of heterozygous plant. That means you want to know the number which is big R, small r. So you only want to know big R, small r. Okay. So the big R, small r is represented by the 2pq. 2pq is the frequency of big R, small r. So students should show that to find out the number of heterozygotes, you need to calculate the 2pq first and then convert to the numbers. Okay. So using the equation, you're able to find out 2pq because once you find out q, you will be able to find out p because p is 1 minus q. Okay. So you calculate 2pq to get this number, you get 1 mark. Then finally, what is the total number for 2pq? Is 2pq times the total number because 2pq is frequency, right? So times total number. So that will give you the answer must be either 511 or 512 only if the calculation is correct. Okay. So B part 1. The B part 1 is a bit trickier, but some of you have done it before, right? It's a passive question. Okay, so let's look at it. So this color of the flower, right? There are 20 different color. Okay, including the shades of blue, purple, red, pink. That means to say other than blue, purple, red, they got different shades, means different darkness. Okay, shades means darkness of purple, blue, red, pink. So that means the color change gradually from one phenotype to the next one. Just like our height change gradually. You got 157, 158, 159, 160 cm and so on. So it's a gradual change. So, and then it's controlled by at least 4 genes. So it's not only one gene, 2 alleles that control, there are altogether 4 different genes that give the color. So this is called a multiple genes are code for the color. And then also the flower color will fluctuate because of CO2 concentration in the air. So these are very important information for you to answer this question. So the students say that the flower color phenotype in this plant is a continuous variation. So the phenotype of flower color is a continuous variation. Suggest 2 reasons why the student make this conclusion. So for continuous, we learned that it must be quantitative, but in this case, it is not quantitative, it's qualitative. Color is qualitative, but there are some other features that fall within the definition for continuous variation in the phenotype. So let's look at the answer. So why is it continuous? Because number 1, the color are not distinct, not discrete, or it is overlapping, or it is arranged, it's a gradual range, or it's a change. So it's not decreased like, ABO blood group is either A, B, AB, and O. Whereas the color is pink or shade of pink, darker shade of pink, lighter shade of pink. So it's gradual change, it's not distinct. And number 2, it is polygene. It's coded by polygene, means more than 1 gene. As long as more than 1 gene is called polygene. In this case, it's 4. Okay. And number 3, the the flower color is affected by CO2 concentration. For example, human height can be affected by the nutrition that an individual obtain, whether enough calcium or not, okay, whether enough exercise or not, enough sleep or not. So in this case, color also affected by environment. Whereas ABO blood group is not affected whether you got enough sleep, whether you exercise, whether you got enough calcium, you will still get your original blood group. So that is called the discontinuous variation, not affected by environment. Okay, continuous variation is affected by environment. So this is an example that they give in the question. So they don't ask directly, but they give you a scenario and you identify from the scenario, is it continuous or discontinuous variation. So here, so they show you the distribution of the color, right? The color is a continuous change from white all the way to black. This is not flower color already. This is what? Suddenly they change to mice. From flower become mice. Okay. It's no longer flower. It's mice color. Right? So originally, the optimum phenotype is the mid brown color here. So this original optimum. So let's look at the question. Suggest why the mice with white fur and the mice with black fur were present in the lowest numbers in the figure. So the reason is because they tell you that the soil color. Okay, let's look at it. So the white and the black color mouse, they are poorly camouflaged. So the one with the largest number is because the color of their body matches the color of the soil. So they're very well camouflaged. So when they're not well camouflaged, what happened? They die because of predation, so their number becomes small. Okay? So this white color, right? Mouse with white color and black color, their numbers are small because the background soil color is mid brown color. So their color white and black against mid brown is not well camouflaged, easily seen by predators, so easily removed, so they die, so their number will be smaller. Okay, so that's the answer. So next question. Next question. So part 2, an environmental event caused the color of the soil to change to dark brown. So now the soil becomes dark brown color. It could be fire or something that caused the soil to change color. So you need to sketch a curve to show the distribution of the fur color in the mouse population after many generations. So that means the soil color is dark brown. So the phenotype of the mice body color selected should be what color class? Dark brown, isn't it? So dark brown becomes the new optimum. So therefore, what you need to draw here is dark brown is the optimum color. Give me the red color. Red color. So dark brown becomes the optimum color. So the rest, the number must drop. So you should draw something like this. But it should end all the way in black. Some students don't get the mark because you end outside black color. Means suddenly there are more black color appears here. So this is wrong, right? Because origin don't have other colors. So it should be, optimum should be at dark brown. And then the curve should be narrower than original. So that's the marking point. Okay. Okay, next question. So part 3, explain what is the meaning of genetic drift. Okay, genetic drift according to the syllabus. So genetic drift, where is it? Okay. Go up. Okay, so part 3, So part 3 is what? Genetic drift is a random change in keyword allele frequency. Okay? Random change in allele frequency. That means this change is not because of natural selection. This change is not because of natural selection. It's because of some events that occur not by natural selection. Something suddenly happened. Okay? So that the alleles, this one is, I don't like this answer. Okay? So number 2, to a smaller population. So that will cause a genetic drift. Something happened to a population, to a small population, causing the allele frequency to change. Okay? So some of you write examples. If I want to mark strictly, I will not give a mark because it's a definition question. Definition question means they don't ask you for example, they ask you for the definition of term, like you open the dictionary. So this word means what? So you need to give exactly the meaning of the word. Okay? So I have marked example. If your example got allele frequency and small population, I also give you correct. Because some students write bottleneck effect, right? Some students write another, what is another one? Founder effect. But actually, strictly speaking, no mark one for these two. You must give the definition. Okay. Next question. Okay. So number 7. So we have got 2 types of organism. One is the American badger and one is black-footed ferret. So these are the 2 species that are found in America. Okay. So this is the IUCN red list for these 2 species. Okay. In 1987 and 2013. So comparing the 2 different years. So black-footed ferret was extinct, I think extinct in the wild in 1987. Okay, in 1987, we cannot find the black-footed ferret, don't have. Okay, after the conservation in 2013, now in the wild, we can find them, but they're still endangered. Last time don't have, means in the wild, you cannot find them. Only in captivity means we keep them in the zoo. Okay, so captive we can find. Okay, so that means extinct in the wild. So question asks a student, so American badgers and black-footed ferrets are both predators. So these are very important information. So American badgers feed on prairie dogs and a range of other animals. Black-footed ferrets only feed entirely on prairie dogs. American badgers do not have any animal predators, don't have. Okay, whereas black-footed ferrets are preyed upon by American badgers and several other predators. So if you were to draw the food chain for 2 of them, right? So this is, let's say I draw it here, okay? So this American badgers, and then we also have the black-footed what? Ferrets, okay? So American badgers feed on prairie dogs and other animals. So we have got prairie dogs here. Prairie dogs. And we have got other animals. Animals means we've got animal A, B, C, and so on. Animal A. Okay, we've got animal B and so on. Okay. So using the food chain, American badgers feed on animal A, animal B, and also prairie dogs. So a lot of supply. Whereas black-footed ferrets only feed on prairie dogs. And black-footed ferrets is also consumed by American badgers. So this is the food chain. So The question asks a student, suggest reasons means a few reasons. Why black-footed ferrets are endangered species, but American badgers are not. So your answer must base on, they only feed on prairie dogs, okay, versus American badgers feed on 3 of them. So the supply is larger in variety, the supply is less here. Then black-footed ferrets can be consumed by American badgers, which would reduce their numbers. Okay, but American badgers got no other carnivores that feed on them. Okay, so let's look at the mark scheme, how you write the answer. Huh? So basically it's the similar answer. Okay, ferrets can feed on prairie dogs or one type of prey only, whereas badgers can feed on a few types of prey. So number 2, when the prairie dog number decreases, the ferrets will also decrease. Remember this graph, huh? Okay, so this is time. This is a population size. Okay, population number. So we have got the prairie dogs. So this is the dog. Huh? So the number is high. So when the number is high, then so what I want you to see here is, this is the predator and this is the prey. Okay, there's a fluctuation in the population size between predator and prey that are interrelated with each other, right? Because the predator depend on prey for food. So the prey number, when it's high, the predator number will be high. When the prey number drops, the predator number also will drop. Okay, so that is about this question. Okay, next question. This question is when a lot of students can't really give the correct answer. Okay, so because why? Because a lot of students give a general answer to the question. Now you see, whenever they ask you general question, suggest how conservation can be done, then your answer will be correct. But over here, they give us a scenario. So in this scenario, they say that, so these 4 agencies, they need to collaborate with each other, and their aim is what? Their goal is to produce young black-footed ferrets to be released into the wild is their goal. So these 4 agencies work together to achieve this goal. So it's a specific scenario, so your answer must cater to this particular scenario, not in general. So the question asks the student, outline how these different agencies could contribute successfully to the conservation of black-footed. So it's not about conservation, it's about what activity they can do to achieve this goal. So the conservation in this case is to achieve this goal, this scenario. So the best way to write your answer is you write what local government do, so 1 mark. What university can do, 1 mark. What zoo can do, 1 mark. What native tribes can do, 1 mark. Okay, each one of them you write 1 to fulfill that goal. So altogether, you can get maximum 3 marks already. It's very easy to get. Okay, but you must write your answer to fulfill the question. So this is the mark scheme answer. So the local or authorities government is the one that will give the funding for education. So if you write funding, you must write funding for what purpose? You cannot write funding for conservation because the question is about conservation. So you need to be very specific. Conservation in what kind of activity, right? Correct or not? You cannot just write conservation because question already say conservation. So to do what? To provide education like campaign, like advertisement, posters, education, all need money. okay, to pass the protection laws, okay? And then university, what do? University will carry out research. Then you give example of the research. How is the research to increase their numbers and to release them to the wild? So to increase the number, we have to improve the success of breeding program, like a research on doing IVF, research on monitoring genetic variability, because when you want to cross them, we make sure that the two parents are genetically variable to reduce chances of inbreeding depression. Okay. Determine suitable habitat. So there are people that can send to go and study different habitats available, which are more suitable for us to release the animal. Okay. And then if we release, we monitor the wild population after release. The numbers maintain, increase, or decrease. Okay. So that is what research can do. Then the zoo is to do what? Zoo is to keep them and breed them. So some students only say, keep them safely in the zoo, and if you say keep them safely in the zoo, I cannot give mark. Strictly speaking, I cannot give mark because the aim is to release them in the wild. So we have to increase their number by breeding them. You see that? How to write your answer to score the full marks. Okay. So if I want to mark strictly, then I need to reduce the marks already. Okay, because I close one eye and mark this question, or else some students get 0 mark. Okay, so Native Americans, what do? Now, the question already said they have got the land, they got reserved land. Okay, so what do? They can provide the reserved land as a suitable habitat to release the animal, okay, to the wild. So this is what they can do, according to question. So please, you need to see what the question is and answer the question. Okay, so next question. So they give you a graph. Now this graph, I already sent telegram to you, right? A lot of students read the graph wrongly, yeah? Correct or not? You look at these 3 lines. These 3 lines is different one, They are 3 different. The first line, South Dakota is solid line, right? Warming is dotted line. So dotted line starts from here. Only warming starts from here, right? Then Arizona is dashed line, right? Start from 1996. This question also same thing. A lot of students didn't answer the full question. I still give mark. Strictly speaking, if I mark strictly, I have to, you cannot get the marks. So let's see how to get the full marks, okay? Describe the patterns of the population growth at the 3 locations, 3 marks. So how to get the 3 marks? You need to describe the pattern of population growth for each of the 3 locations in full. You must describe fully. Some students only choose, only here I describe. Cannot. Okay, if the curve starts from here, you have to describe from here until the end. So that's when a lot of students didn't do that. They selectively choose anywhere and then they answer the question. Cannot. You must answer the question for South Dakota. From the beginning is 1996 all the way to here. Hello, why got no line? From here all the way to here is for South Dakota. Warming is from here all the way to here. I have to describe the whole pattern. Arizona from here all the way to here. So this is how you describe. Then one more mark is to quote the figures. So there's more than enough points to score the 3 marks. Okay, so let's look at the mark scheme. So South Dakota is the first one. It increased steeply. Number 2, warming. Warming initially was constant, right? The curve didn't change. You see not? Initially here for first few years are constant. So you need to write is constant, right? After constant, then it will decrease before increasing. So this is called the pattern. So it decreases before it starts to increase. So you need to describe the whole thing. Very few students do that. Very few. Maybe 1 or 2 only. Okay. Third marking point, Arizona population start at very low level, then increase. So you can say Arizona population starts at very low level, fluctuates, and then increase. Okay. Then fourth marking point is you give comparative figures. So at the same site, you compare between 2 years. That means to say that warming, okay, I compare at the year, I compare this year at the beginning versus this year in 2004. Okay, same location, 2 years, or you can compare 2 locations. So I can compare South Dakota, warming, and also Arizona in the year 2004. What is the total number? So I can compare these 3 locations. In a particular year, I can compare 1 location in 2 different years. Okay? Okay. So next question. Is everyone okay? Yeah. Next question. Now, next question is quite easy as long as you answer the question, okay? So there's a data here that shows what? Table 7.1 shows gene pool data and lake size. Okay. This is percentage of polymorphic genes, mean number of alleles. This is mean length of lower back leg bone, mean length of lower front leg bone, okay, of South Dakota, Wyoming, and Arizona. Okay, this is a very important information. The population at South Dakota in 2004 maintained the same level of genetic variation and leg size data as the original captive population. So all these 3 locations, right? The black-footed ferrets in all these 3 locations come from the same original captive population. And then from here, they are released to these 3 locations. Okay, so after release, in 2004, they obtained this data. Right, in 2004, they obtained this data. They say that South Dakota data, which is this one here, all these numbers remain the same as the original captive breeding population. That means this one same as original. So therefore, for Wyoming and Arizona, we have to compare this with the original one to see whether there's increase or decrease. Okay, same. So we compare these 2 with this one. Okay, and see whether there's an increase or decrease. So let's compare here. So we see that this is a decrease here. There's increase for this one. Okay, you compare with this. Then for this, you compare, there's a decrease here. There's a decrease here. Whereas this one is the same. Okay, this one is the same. Right, this is the same. There's a decrease here. Okay, so after compare, then you can answer the question. So use a table to describe how the gene pools and the leg size of the Wyoming and Arizona population have changed compared to original. So very easy. Gene pool means anyone from here, increase or decrease. Leg size means everyone from here, decrease or increase or decrease. So Wyoming, gene pool decrease. Okay. And then Arizona, mean number allele per gene pool increase. Then leg size, Wyoming decrease, Arizona remain the same, so no need to say because it didn't change, no need to write. So that's the mark scheme answer. Okay. Can So number 8, define recombinant DNA. So recombinant DNA, I've given to you in your notes that it is a DNA, right? Artificially produced by joining two DNA from different sources or organism. So this is the definition I've given to you in your notes. Right? It must be a DNA where it's artificially produced by joining or combining two DNA that come from two different sources or two different organisms. So definition, you need to memorize. Okay, so please memorize the definition. Because you can see in this question itself, they ask you a few definitions. Okay, like genetic drift, recombinant DNA. Okay. Okay, next question. B. This plasmid includes a gene that codes for fluorescent protein GFP. Explain the purpose of including a gene for a fluorescent protein in the plasmid. So when you say that the protein is a marker, is not correct. Marker is the gene. Marker, this word denotes the gene. Okay, so this is a common mistake among the students because even the textbook also didn't explain clearly. I realized because I reread the textbook. They didn't specify very clearly that the marker definition is actually the gene. Okay, so let's look at the mark scheme. So that is a marker gene. You see, not marker is a gene. So that is a marker gene. It is used to identify transformed bacteria. That means bacteria that has taken up the recombinant DNA. Okay, so how do you identify it? We have to detect it by using UV light because it can fluoresce under UV light. Okay, so what is the purpose of the gene? The gene's function as a marker in order to identify whichever bacteria that's taken up the recombinant DNA. So how to identify? When you shine UV light, the bacteria that's transformed will fluoresce. Okay, so next question. Do you have a class afterwards? Yeah. Okay, so should we stop here? I think we should stop here. So you stop here. Whatever we cannot finish, we'll continue in extra class. So we'll decide when we can do extra class. Do you prefer nighttime or weekends? Nighttime. Or who prefer daytime? Daytime. Daytime cannot because your Wednesday, Thursday, Friday, all your class until 5 p.m. Your workshop, hello, your Wednesday, Thursday, Friday, you must come for class. You got workshop to attend, face to face. Okay, so who prefer nighttime? Who prefer Saturday, Sunday? So majority prefer nighttime. So I will see which nighttime you can. Is 8:30 okay for you? 8 o'clock too early, I think 8:30 is just fine. Today. Not today lah. I will decide which day, okay? So I'll decide and text you all lah. So that's all. Thank you very much.